Welcome to Study Loop.
Okay, so problem 40 here is really just sort of synthesizing information from this, what looks like a sort of made-up experiment, right? And so they have some dividing cells and they say, stop dividing, okay? So they get all the cells to stop dividing and then put it back into a new medium, which releases the box and they can start to divide again. So it gives them all this sort of like, they're all starting at like a fresh starting line rather than sort of all possibly being in the middle of cycles and whatnot. So we take our first sample, sample A, right, and we get a relative DNA content. So it's relative. So this is, since this is the first sample, its relative DNA content is 1 because we're sort of comparing it to the first sample, right? And then we move on to the next samples, and the next samples all have a relative DNA content of 2, right? So that means it has double the DNA content of A, right? Because A had 1 and these all have 2. Okay, so what that means is when we're looking at the DNA content of B relative to A, it has 2 times the DNA content. Okay, so it has essentially gone through the entire sample, possibly 1 cell division, right? It's now has twice as much DNA. So the answer here is going to be B, that the DNA content has doubled.